In this lecture, we discuss cellular structure. Most animal cells have three basic components. They could have more. In fact, many animal cells do have more, but they at least have these three: plasma membrane, a cytoplasm, and a nucleus. Now, the plasma membrane provides several functions: a structural support, so it provides some shape for the cell. It allows for communication with other cells, and it allows for identification of itself. It also helps us define the intracellular versus the extracellular fluid. So intracellular fluid would be on the inside of the cell. Extracellular fluid would be on the outside of the cell, and the plasma membrane would be the dividing line. Now, our plasma membrane is made of phospholipid bilayer. We have one phospholipid with a hydrophilic head, or a polar head, and hydrophilic or non-polar tails. Because we have two different properties, polar and non-polar, or hydrophilic and hydrophobic, whatever terms you prefer, because both of these characteristics are on the same molecule, we refer to this as an amphiphilic molecule. So we take these phospholipids with their little hydrophilic heads and little hydrophobic tails, and we arrange them in this bilayer. Notice that the heads on both parts of the layer are facing the intracellular and the extracellular fluid, which is full of water. But coincidentally, the hydrophilic polar heads are perfectly okay with that. You will notice that the middle of the membrane is full of those hydrophobic fatty acid tails, and you will also notice that there is no water in here. This is by design. Now, we don't just have phospholipids in our membrane. We have other parts as well, as you can see in this image. Because we have multiple different parts, we refer to the membrane as a fluid mosaic. Fluid because it is dynamic. We are able to move this. We can get things in, we can take things out of the cell. We can even move things through the bilayer itself. We could go through a protein channel instead. Some of these phospholipids can move a little bit. There's some fluidity here, and that's important as well. Now, speaking of all these proteins, we do have a couple varieties of proteins that are found consistently in our plasma membrane. We have integral and peripheral proteins. Integral proteins, which are sometimes referred to as transmembrane proteins, will go from one side of the cell all the way to the other side. Okay, spans the entire plasma membrane. While peripheral proteins, on the other hand, so this little guy, for example, is only found on one side of the plasma membrane. So this guy's inside, this little guy would be outside. But again, notice they're only on one side. We've got our examples, integral and peripheral proteins here. You will notice the little phospholipids. We have a ton of those guys. We even have cholesterol, some carbohydrate chains that serve a few different purposes. On the inside, right beneath the membrane, we have some cytoskeletal filaments that help with the structure of the cell as well. So again, the idea of the fluid mosaic meaning multiple parts, is really illustrated well in this image. Now, we mentioned the integral versus the peripheral proteins. Let's talk about some of the functions of those proteins. Some proteins function as transporters. We have two main categories: protein channels and carrier proteins. So protein channels move substances across the membrane. So, for example, this is kind of like a wide open door. So as long as the door is open, our substance, our solute, is going to be able to pass through very easily. Our carrier protein works just a tiny bit different. You now have to have the substance bind directly to the carrier protein before it is transported. And so notice that our solute here has to bind to our carrier protein before the carrier protein opens up and allows that solute to pass into the cell. So just a tiny bit different there. Now, some proteins are receptors instead. When these receptors are activated, they trigger events inside of the cell. One of the ways that we trigger these events is through the binding of ligands, which are chemical messengers. And so in this example, we have our little ligand, our little substance, and it binds to our receptor, okay, receptor on our protein channel. And once that happens, notice what the protein channel does. So once we have bound, this protein channel opens up, and now we are allowing solutes to come into the cell. Okay? It wasn't until the ligand, the messenger, bound to the receptor, that was when we could open up this channel and allow for the transport of these solutes. Some of these proteins work as enzymes. We have mentioned that enzymes speed up chemical reactions by lowering activation energy. So in this example, this big green enzyme is bringing these two substrates together. We are combining them to form one product. Okay. Some of our proteins are structural for support. These tend to be part of our cytoskeleton. Okay. You can find these outside of the cell, but they're very, very common right on the inside below the plasma membrane. And then we have cholesterol. Let me see. Yep, here's our picture. This little tiny molecule, look at it, it's really tiny. This is cholesterol. Okay. Now, we have mentioned cholesterol. It is a lipid molecule. Cholesterol usually gets a bad rap. Your doctor is always telling you, oh, you got to lower your cholesterol, and that's all true, but not all cholesterol is bad. In fact, some cholesterol is actually vital to your existence. One of the functions of cholesterol is used as a starter to create steroid hormones, which are important, but for our discussion here, we use cholesterol in the plasma membrane. So again, there's not a ton of them. You've got one here, you got one here, you got one way down here. This allows for your plasma membrane to stay fluid during temperature changes. So if your cells got really, really, really cold, when you get really cold, molecules tend to pack closely together. If we pack things too closely together, that could negatively affect our ability to transport molecules, and we don't want that. And so cholesterol helps prevent everything from getting a little too close, and so it does not negatively affect transport between our cells. Now, last but not least, we have glycolipids and glycoproteins. These tend to be on the surface of our cell membranes. These are used to help identify our cells. So for cell recognition, we need to be able to recognize ourselves for proper immune function. So your body should be able to recognize your glycolipids and your glycoproteins. If your body cannot recognize your cell parts, then that's where we start to get autoimmune disorders, and we all know that those are not really a good time. Okay, so that was the membrane. Now we're going to do our cytoplasm. Your cytoplasm is technically a couple of parts combined. You've got your cytosol, which is your intracellular fluid. This is mostly water, but there are some dissolved solutes in your cytosol as well. Now, inside your cytoplasm, you also find your cellular organelles. Each organelle has a very specific function. We call this compartmentalization. So it's kind of like, so if we think of our organelles, each of these little colored structures, if we think of these as, just for example, little boxes. Each of these little boxes is its own separate little box, and We do that because sometimes the interior, whatever's going on inside of these organelles, sometimes there's a very harsh environment in there. So for example, some like lysosomes, for example. The internal environment of a lysosome, it's very harsh because lysosomes, their job in life is to destroy things. We want to keep that environment separate from the rest of the environment of the cell because we don't want to break down the whole cell. And so this idea of compartmentalization allows us to do some very specific functions in a very safe way. And our last component of our cytoplasm is our cytoskeleton. We've briefly mentioned this. We have our protein filaments that allow provide us with a shape. It gives us some strength. It also allows for transportation. Okay, it allows us to move organelles and substances within the cell itself. And let me see if I can get you a good example. Let me go back. Aha, here. Do we remember these structural proteins that I mentioned a minute ago? This is part of our cytoskeleton. Okay. Okay. All right. So we just mentioned the word organelle. Okay. We said each organelle has a very specific function. These come in two categories. We've got membrane-bound organelles and non-membrane-bound organelles. So membrane-bound organelles are the ones that are going to want that very specific internal environment for whatever reason, and so we wrap it in a completely separate membrane. You've got your mitochondria, your peroxisomes, your lysosomes, your endoplasmic reticulum, or your ER, and your Golgi apparatus. All of those have an additional membrane. Now, your ribosomes, your centrioles, cilia, flagella, okay, those remaining organelles do not have an extra membrane surrounding them. Now, we'll go through some functions. Your nucleus, okay, so your nucleus is where your DNA lives and your RNA. And we've really briefly chatted about DNA and RNA already. We know that not only does your DNA make you who you are, but it contains the instructions for protein synthesis. RNA is the actual instructions on how to make your proteins. All of this DNA lives inside the nucleus itself. Most people have at least heard of the nucleus. They're somewhat familiar with it. This is one of your organelles that does have an additional membrane as well. We call it your nuclear envelope, or perhaps your nuclear membrane, whatever term that you prefer. You will notice in our images here, there are holes. Okay, all these little dots are holes. These are pores. These are called pores. You are making mRNA and you are making ribosomes inside your nucleus. We're going to need to get those out of the nucleus and into the rest of the cell. So what do? We send them through those little pores. Okay, so those are by design. Kind of looks like a little golf ball almost. You will notice in the middle this really dark spot. This is called your nucleolus. Okay, sometimes that's a little hard to spell, that extra L in there. Your nucleolus is where your ribosome components get made, and your ribosomes are going to be responsible for protein synthesis. So that's one extra little guy that actually does play kind of an important role. Okay, so the rest of our organelles that we need to be familiar with. Your centrosome. Okay, so this little contraption up here. Your centrosome is composed of centrioles. This is used during mitosis, also known as cell division. Okay, so when we go to take this one cell and turn him into two cells, your centrosomes are going to be what helps pull those two cells apart. Peroxisomes are used for detoxification of potentially toxic substances in our bodies that we want to get rid of. We just mentioned your nucleus contains your DNA. We also just mentioned that your nucleolus is where ribosomes are created. Your rough endoplasmic reticulum, your rough ER, this is where protein synthesis actually happens. The rough part of this rough ER is coming from the ribosomes themselves. So this image, and that might be a little small for you to see, but if you can see all these little red tiny dots, those little red dots are the ribosomes themselves. This is where protein synthesis is occurring. You also have a smooth endoplasmic reticulum, or a smooth ER. It's smooth because it does not have the ribosome, little dots. Instead of making proteins, we're going to make lipids here. So for example, the phospholipids for your plasma membrane get created here. We mentioned your cytosol is part of that is your intracellular fluid. Lysosomes are good for cellular digestion. Okay, so we could digest old and worn out organelles. We could ingest a bacteria and need to break it down, all kinds of good stuff. It's kind of like a garbage disposal almost. We've mentioned your plasma membrane several times. It helps us separate inside from outside of the cell and has several other functions as well. Your Golgi apparatus is kind of like FedEx or UPS. It modifies, packages, and ships proteins. So once you create a protein on your rough ER, you send it to the Golgi. It modifies it, makes sure that it is fully functional, and it packs it and ships it off to wherever it needs to go. Some proteins stay inside of your cell to function, but some proteins will actually have to leave your cell to actually go do their job somewhere else. Now your mitochondria, this is the site of cellular respiration. This is where we make ATP. Okay, this is the site of ATP production. Ribosomes, we've already mentioned several times. This is where actual protein synthesis happens. Some ribosomes are bound to your rough ER. We've already mentioned those. We kind of pointed those out, these little red dots. And again, I know this might be a little hard to see, but you also have some free ribosomes, these additional little tiny, tiny red dots. The types of proteins that are made in these two separate ribosomes, they just have different functions. They're still making proteins, they're just different kinds of proteins. And last but not least, we have vesicles. These are for transport. You can create something, put it in a vesicle, and ship it off, and it'll leave the cell, and it'll go somewhere else and do its job. Or we could bring something into the cell, put it in a little vesicle, and take it inside the cell wherever it needs to go. So they're almost like tiny little boats. All right, we've mentioned the cytoskeleton a couple of times, just kind of reinforcing. These are made of protein filaments, which provide strength. We can also do cellular movement with motor proteins. So in this example, we have taken a vesicle and we would like to move it around in the cell. So we attach it to this kinesin motor protein. And if you can imagine these little blue things as feet, this protein will literally walk across this microtubule, and it is going to use ATP, but it's literally going to walk across this microtubule, and we can move the vesicle, which is pretty cool, to be honest. Motor proteins or protein filaments are also involved in phagocytosis, which basically means we're bringing things into a cell, and even muscle contraction. So actin and myosin protein filaments are what actually make your skeletal muscles move. So this is all pretty cool. Look at this little guy. Look at his little feet. He's just walking. Okay. All right. That